Welcome, everyone. I know there's still some people coming in and getting settled, but just to stick on schedule. So good morning and welcome back to Track 4, Science and Technology Innovation. We hope you enjoyed your morning break, sponsored by EMD Serrano, and are ready to dive right into our first breakout session. My name is Tammy Johnston from Ambry Genetics, and I'll be your host for these morning sessions. Once again, we would like to remind everyone that we are live streaming, and I would like to welcome our remote attendees to the session. As a reminder, we have our mobile app available. Be sure to download it to use some of our highlighted features like live Q&A. Without further delay, I would like to introduce our first session of the day, Making Sense of Antisense and Other Oligonucleotide Therapies, presented by Sarah Boyce, Chief Business Officer of Ionis Pharmaceuticals, and Molly Harper, Vice President of Commercial Development at Exia Pharmaceuticals. Please join me in welcoming them, and we'll begin with Sarah.
Firstly, thank you so much for that introduction. And I see rounds of applause as people recognize themselves already. It's a pleasure to be here. And I'd like to thank the organizers at Global Genes for hosting this incredible conference for us all and for the opportunity to try and make sense of antisense. I am not a scientist. But I am someone who has a deep love and deep passion and respect for what tremendous science can do. At Ionis, we feel a very deep responsibility in that we have a technology in our hands that can provide solutions to many different diseases that are not really applicable to other types of technologies. And I'll explain why. And it's why when we have our slides, we have the patients that we serve that are front and center, and some people might recognize themselves during this. Just want to introduce to you this gentleman here. His name's Charles Sabine. Charles is one of the most eloquent, charismatic people you could possibly meet. He's an ex-news correspondent for BBC, so you may have guessed from my accent that I'm also from the UK. And he has Huntington's. Huntington's is one of the most devastating neurological genetic diseases and is today untreatable. There are no approved therapies for the treatment of Huntington's. And at Ionis, we do have a drug that's currently in phase 1/2 for Huntington's. It's called Ionis HTTRX. And one of the aspects is Huntington's is the type of disease that's really unique for Antisense technology because there is a very precise target. Antisense, we work on the RNA, so the DNA is left alone, the protein production is left alone, and we kind of get in the middle of it. And the goal is, in the case of Huntington's, where because of a repeat of a toxic gene, these patients overproduce a toxic protein. The protein gets stuck in the cells and the cells die. So we're hopeful and very much cheering on the program along with all of the patient community. But that's the type of work that we do at Ionis. And our goal is quite simple. It's to revolutionize medicine and save lives, which is a pretty audacious goal, but we're going to give it our go and give it our best shot. And special shout out to Justin here and his beautiful wife, Naomi, who are celebrating their 15th wedding anniversary today. So... and then also in terms of, so Ionis was started 26 years ago with the goal of developing a new platform for drug discovery and development, which is antisense technology, and to do that using RNA. And then we also have my colleague here, Christina Boyer, who's over there. Christina is our head of patient advocacy, but also, even more importantly, is one of the founding scientists. So she started with the company 25 years ago. And for Falada Source Sin, which Molly will talk about later, which is a drug we have now in development and registration for FCS. Christina's name's on the patent, so she's a bit of a rock star here with us today. Yeah, she started when she was like 5. So what is antisense technology? So really what we're doing is using RNA, and we have our molecules are a single strand, and essentially they interact with the RNA that comes from the DNA to do a range of things. That may be things like we're looking to change the splicing, so to have something skipped, or to bring 2 pieces together where we can then get a protein produced that actually shouldn't be produced at all. It's an amazing trick of almost mimicry where we can get the body to produce a protein that technically it shouldn't be producing. We can also do things like we can act on microRNAs, we can act on structural RNAs, on things called non-coding RNAs, which actually have an important role in cancer. So there's a whole range of different mechanisms that we can interact from a technology perspective. From a specificity perspective, one of the other unique advantages, it allows us to be incredibly quick. So I joined the company 3 years ago, and I think one of the things when I joined that really blew my mind was the aspect of the speed. So essentially we can go from target detection or target identification, so that's identifying disease, identifying a genetic target, to actually getting a drug ready for IND-enabling tox and into man much faster than anybody else. Now, this is the path if everything goes to plan. I mean, typically, our time frame is a bit longer than this. It's more like about 18 months. But typically, we're 12 to 18 months to get to IND-enabling tox to then to be planning those in-man studies. It allows us to have incredible pace. We put about 5 to 6 drugs into our pipeline each year. One of the reasons why we can do it is because it's a platform. So all our drugs are using the same type of technology. So from a chemistry perspective, the chemicals are essentially the same. How they're arranged is different. And that's how we will have different approaches. But from a chemistry perspective, they're all very similar, so they behave in very similar ways. So it allows us to kind of know exactly which experiments to run and why. I talked about this earlier. So essentially, we come in here. So the DNA is left on its own to do its thing. And essentially, whatever that protein is, we affect it by acting here, whereas typically for monoclonal antibodies or small molecules, they tend to interact here, whereas we're sort of more upstream. I say to my kids, it's like, we change the message, we change the email, so to speak. And it's from one of the limitations of small molecules and antibodies is they need something to attach to. So they need something to attach to on a protein. They need a receptor on the cell. They need some sort of interaction point. By moving earlier, you're then free from that. And that's why there's a lot of diseases that just aren't treatable with a small molecule or a typical monoclonal antibody approach, because there isn't anywhere to attach, or there's so many places to attach that you just can't dose enough drug. So for example, for Lp(a), where there's a lipid called ApoA, there's so much of it in the blood that you can't attach a monoclonal antibody to it, because the amount of antibody that you would need to give would be quite simply overwhelming. This is complex, and I said to Christina, you know what, let's go in detail and explain each pathway, but really this is to show the range of different mechanisms at a biological level in which we can interact. And as I go through, I'll give you specific examples of how we've used that in our technology. I'm going to go back. So just on a basic level, if there's less RNA, there's less protein. If there's more RNA, there's more protein. And we can do either. So we can upregulate or downregulate. And then occasionally we get in the middle and look to really change things like we did in our program for spinal muscular atrophy. And, and the beautiful boy you saw at the start, young Asher, who's just gorgeous. So we can work on all types of RNA. We can work through a range of different mechanisms from a route of administration. kind of done every single way you can probably get a drug into someone's body, but most of our drugs are given through a subcutaneous injection, or in the case of where we have a neurological target, they're done through an intrathecal injection into the spine. That's because Antisense does not penetrate the blood-brain barrier. The blood-brain barrier has an incredible protection mechanism, as it should, to make sure that things don't cross that barrier. So for Antisense, we go straight in for an intrathecal approach. But because we have a long half-life in the spinal, where we can dose pretty infrequently. So for that, we're dosing once every 4 months. And from a tissues that we can work on, it's pretty much broad throughout the body. A lot of our targets are liver targets. That's where we started. But we have a very large neuro program now, thanks to the incredible Frank Bennett, who is our the scientist behind the neuro program and the leader of our neuro program, and our partners at Biogen, who we work with every single day to advance our drugs there. To give you, and this just gives you a range of, so I know this is a rare disease conference, or in many ways with genetic diseases, they tend to be rare, but we do actually treat much broader diseases. When we look to identify a disease and a target, we really look at, is this something that isn't applicable to other drug technology? Because when you have a unique technology, we also feel we have a responsibility as to where we use it. And we choose not to go into spaces where we're competing with other technology, because another technology can solve it, and we have so many places in which we could work. And then also going into very rare diseases, so things like ALS through SOD1, which is a specific mutation in about 5% of patients. We have a phase 1/2 program that will probably read out mid-next year in SOD1. Huntington's, I spoke to a little bit with regards to speaking about Charles, and we're looking forward to the readout from the phase 1/2 study from that program, which will probably be early next year. And then TMPRSS6, which is a different approach for beta thalassemia, which we're just putting into the clinic now, which is a really exciting approach. So in terms of the straight knock down the proteins, that's the case of Falada Source Sin. I'm going to talk a bit more about Spinraza, and then also some of our early science where we're looking at how can we move the technology even further and look at different approaches. And one of those is, an example is targeting upstream ORFs, which is open reading frames. So this is when you start to read the DNA, where we can come in here and change it further up. And by changing it further up, again, you can go up and down, as opposed to specifically addressing a certain target. I can't not talk about SMA as an example. I think I might be lynched by some of the people in the room if I did that. I think, as we know, the treatment of SMA has changed dramatically with the arrival of an approved drug. And in the case of Spinraza, both for all types of SMA, so both for infantile onset and later onset. And the team at Biogen is doing an incredible job at making sure that they can get Spinraza to the patients who need it as quickly as possible. So I must give full credit to the team there, who really are incredibly dedicated with the work that they have been doing. But just to give you a little bit as to the approach and how we chose our approach. So the SMA protein, which is survival motor neuron protein, is essential. It's essential in your body. And unusually, there's actually a backup. So there's the SMN1 gene and the SMN2 gene. The SMN2 gene is a backup gene. So most of us, we all have an SMN1 gene, and the SMN2 gene doesn't really do much. It produces a small amount of the full-length protein that you need, but the majority of it is coming from the SMN1 gene. So for us here, the SMN2 gene doesn't really play much of a role. One of the aspects in looking at how to target this disease, in the case of SMA, the SMN1 gene isn't there. So then the approach is, okay, well, what can we do with the SMN2 gene? And how can we get the SMN2 gene to produce more of the protein that's needed, because you don't have the traditional mechanism. It's a really clever approach. It's a really clever approach that kind of in many ways relies on the genius of your own body's biology, that there is a backup gene to produce a protein that's really important, which then allowed us to really think around what we can do with Spinraza, which was essentially we alter the splicing. So we get in the middle here, and we can upregulate so you can produce more of the full-length protein. When you produce more of the full-length protein, and then when we saw that, firstly in animals and then taking into the clinic, we've really been able to see tremendous change in the treatment of SMA, and where we've been able to increase the protein enough, whereby for pre-symptomatic infants, so this is being able to treat before they're physically showing the signs of the disease, obviously all infants in the study are alive and well and event-free, which isn't typically what you would expect. But the majority of the infants in the study have been achieving their motor milestones at the appropriate age. I saw a video from one of our investigators of a young boy. I think he was 15 months old. And this kid was like running around and like running away from the doctor and climbing on the chair and you would have no idea that gorgeous little boy actually had SMA. It was incredible. For infant onset, we've been able to increase the event-free survival and improve strength and function. And we take incredible joy when we see sort of stories and pictures of infants being able to sit and being able to walk and being able to stand. And then in later onset, we've actually had children who had lost the ability to walk actually regain it, and to increasingly grow in strength. So an incredible achievement for the technology, and Spinraza was approved on the 23rd of December. This little boy here, Logan, he was recently treated. He's absolutely adorable. And Logan, he can walk, but he gets tired, and so he can't walk well. And our hope for him now, he's treated with Spinraza, is we'll be able to get him being stronger and stronger, and where he wouldn't have to face the possibility of ultimately being able to lose the ability to walk. So we're thrilled for Logan and Rosie and the family. So that's in terms of, I guess, where we can create, in many ways, it's an example of our technology at its very best. It's an example of science at its very best, and it's an example of nature at its very best, where there's a backup gene, and the body's smart enough to actually have that. And then when we can design something that is very, very precise. You'll hear a lot about precision medicine, and precision medicine is only enabled by the body itself, and then by deep understanding as to what are the mechanisms from a scientific level to then specifically to be able to design a drug. And just to finish up on the gorgeous Asha, who is one of the children, or one of the infants who's now a child, who was gracious enough, and his parents were gracious enough and brave enough to take part in a clinical trial. It takes tremendous courage to take part in a clinical trial, and we're very, very grateful for the people who do, because it really is a selfless act, and who's continuing to get stronger and stronger. So that's a very quick tour of Antisense technology and why it's different and some of the things that we can do. And I'm going to hand over to my wonderful colleague, Molly Harper, who's going to talk about the work at Exia.
Thank you, Sarah. And I want to start by echoing two comments Sarah made in appreciation. So first is appreciation for Global Genes and having this incredible patient advocacy summit. Exia is very, very proud and very honored to be here for a third time. And it's my second time, and it's always inspiring and just wonderful and invigorating for everything we do. And the other piece, of course, to echo what Sarah said in terms of appreciation for the patients who've been in our clinical trials. It would not be possible to do what we do and bring therapies for other patients without that commitment, and it's humbling for all of us. So building on Sarah's presentation, what she highlighted by starting with Huntington's and then walking through the tremendous example that was seen with SMA are diseases and therapies that began very, very clearly with the end in mind, right? So with SMA and with Huntington's, as she described, it's a very, very clear understanding when they designed that first molecule about the protein, the disease, the gene they were going after. What I'll be highlighting here, which has been the work of Exia recently, is where that science interplay that Sarah described also folds into the development process and how learnings during the development process, in the case of Falada Source Sin, as I'll be sharing, shifted the direction and identified opportunities for rare diseases which were not seen when they originally created the molecule itself. So just some additional background in terms of Exia. So Ionis, as Sarah outlined, began 26 years ago as really the pioneer in antisense therapies. Exia began 2.5 years ago and was created by Ionis specifically to do the late-stage development commercialization of their lipid portfolio, so therapies that were in development for lipid disorders. We're based in Boston. Ionis is down the road in Carlsbad, but obviously we're colleagues and affiliates. Another point, too, just related to this particular slide, is that partnership, and this is so appropriate to be here at Global Genes, partnership is at the core of what we do at Exia, and that's what this slide represents. So on your left is Jamie, who in the Regulatory Affairs Group led the filings in July, August, and September for the US, Europe, and Canada for Falada Source Sin. And on the right, you see Yang, who lives with FCS and is someone we've been privileged enough to meet and who's sharing his disease experience has changed how we've thought about not just the disease and not just us, but actually the broader medical community, but also how the therapy can affect the disease and the science thereof. So again, it's this interplay, science, drug development, patient experience that we'll be highlighting in this example today. Okay, so again, Exia was really formed to bring forward the lipid portfolio of Ionis. And as Sarah explained, the therapies begin with genetically identified targets. And these are the 3 targets that were identified and therapies were originally developed for, or the molecules put into development for. And a key point here is the concept of genetic validation. So what does that mean? So genetic validation I've also heard described as an experiment of nature. And I'm not a scientist either, so the way it was simply explained to me is an experiment of nature is when there's a population that is missing a gene and that's recognized and people understand what happens to them, and that's been proven. So what you see here are these specific proteins and what was published in the literature that showed that they were genetically validated. There was some difference in the people who had alterations in these genes. And what this story is about, the story of changing directions to affect rare disease, you can see we'll be focusing on our ApoC3 antisense, or Falada Source Sin. And originally, it was really identified as aligned with cardiovascular risk reduction, which is still very true. But what's also been exciting is what we've learned during the development process of how it might be able to affect the 2 rare diseases that it's being studied in. So again, Falada Source Sin is an ApoC3 antisense. It's being studied for familial chylomicron anemia syndrome and familial partial lipodystrophy. And on this slide is Ahmed, who lives in London and has also shared his experience living with familial chylomicron anemia syndrome. So ApoC3, this target, it was genetically validated and associated with triglycerides. Triglycerides are a lipid parameter. Cholesterol tests, we're all familiar with them. Usually you have triglycerides on it. And work that was done prior to us, that genetic validation, showed this association. So the organization recognized and the scientists recognized this is an interesting target. Triglycerides are important medically. Let's see what can happen. So they developed Falada Source Sin. And again, I'm not a scientist, but I think these are actually pretty cool drugs. So what the drug actually is oops, sorry. Not a pointer. What the drug is this remarkable sequence of synthetic DNA, only 20 bases long, but it's been designed to find those similar 20 bases in the whole human genome that correspond for coding this protein. So that's the drug that initially went into phase 1 studies. And it wasn't a huge surprise because of the genetic validation that inhibiting ApoC3 did lead to profound reductions in triglycerides. But what was exciting was then what happened during the phase 2 process. I mean, it was all exciting, but what was, a new twist happened during phase 2. So these show some of the publications that arose from the phase 2 studies. So 2 things happened that related to finding opportunities, potential opportunities for 2 rare diseases. First, a lipid specialist, a well-known expert in San Diego, Dr. Joe Whitsom, who worked closely with Ionis, said, this drug is so powerful and so remarkable, and there is a patient population for whom nothing else works, and we should study that patient population. And what this image on the left represents is a unique publication done in the New England Journal of Medicine. It was particularly for groundbreaking science, because what it showed is that this drug worked in this patient population, familial chylomicronemia syndrome, that previous science suggested it shouldn't work because of the way people had previously understood the science. At the same time, scientists and expert physicians who work on partial lipodystrophy said, you know what? ApoC3 and triglycerides are important in this disease. It's a different type of disease. There's a lot more going on beyond triglycerides, but this could be important. And at the same time, a study was presented that showed ApoC3 is an important factor in lipodystrophy. That led to a phase 2 study on insulin sensitivity and the potential for Falada Source Sin to affect insulin sensitivity, which is the core of partial lipodystrophy. And it showed potential for beneficial effects there. Both very exciting, both totally different from the original intention of the drug when it went into phase 1 and when it was originally designed. So again, a difference in how the process of development itself, partnerships with medical experts, partnerships with doctors who really understood these patient populations can create new paths for drug development and opportunities to help different patient populations. So familial chylomicronemia syndrome, why was there such a need? High triglycerides, there are many therapies available for high triglycerides. There are many therapies available for cholesterol. Well, FCS is a little different. FCS is actually a deficiency in an enzyme, lipoprotein lipase, like so many other diseases that are represented here at Global Genes, where there's an enzymatic challenge, a deficiency, either not enough, inability to make it. There are many different mutations that can contribute to FCS, some of which have not been characterized. But again, this problem with this enzyme means that they can't break down chylomicrons into fatty acids, so there's the buildup. And chylomicrons, when they are in excess, can lead to triglyceride levels 10 times, 20 times the levels of normal. And that's significant because of the sequelae. Ultimately, there's a risk of potentially what can be fatal, which is acute pancreatitis. But then, as we've also learned, significant daily symptoms and manifestations that truly affect people's daily lives in very profound ways. But again, why was there an unmet need, right? Again, there's so many therapies for lipid disorders. The issue is that the conventional drugs for lipid disorders work on this enzyme, which is not functioning in this particular disease. So there needed to be a new mechanism. Remember, as Sarah described, Antisense, the vision of Antisense and the idea of it, and what is so exciting, is it's a different way of tackling problems that are not druggable through other means. So that was interesting. Another question we often get is, if this is an enzyme problem, why can't we make an enzyme? That's what enzyme replacement therapies are. Could we have an ERT for this disease? What we've been told by the scientists is that this particular enzyme is such a problem with its half-life, it's so quick that it's not viable for that particular technology. And it's also not viable for gene therapy because it's not a pure single gene disease. So again, that idea, it's a problem mechanistically as a disease that has not been druggable through other technologies. And Antisense has the potential and has shown the potential to help and reach the targets. I guess there's a build. Okay. So as we started exploring this therapy for this disease, we also started learning more about the disease because we were thinking about how we're going to communicate to regulators. What is the data we're going to collect? And again, in that spirit of partnership, and we're privileged here to have members of the S community here who've been generous enough to share their experiences, we've learned so much more about the medical complications, the medical burden that people live with. In addition, that partnership also has allowed us to quantify the aspects that are both medical and also other factors. For example, effects very significant in terms of ability to work full-time jobs. And this is an example of new data that we've been able to bring to the medical community that they've found incredibly remarkable new learnings. And it's a function of, again, that interplay between the science, drug development, and the spirit of partnerships with physicians and the patient community. So earlier this year, we were privileged enough to share, and really excited to share, the results from the pivotal phase 3 study for Falada Source Sin in familial chylomicron anemia syndrome. So as I discussed, this is a disease that's characterized, and the primary endpoint for the study is triglyceride levels, which were typically the entering triglyceride level of the study was 2,000. For comparison, for most people here, or the average person's triglycerides target is around 100. So again, 20 times what is normal or desired. So it was very exciting to see these results in our pivotal phase 3 of a 77% reduction. Tremendously exciting, the first study ever of its size, and the first to show a therapy with this potential benefit. So since then, we have moved on to file, as I'd mentioned, in the US, Europe, and Canada. In addition, we were able to assess additional endpoints, which really capture, of course, the endpoints and the considerations that are most meaningful to patients and to physicians. Because of the study size and the challenges, these weren't primary endpoints, but we were able to see changes in reductions in the incidence of pancreatitis, pain, both the frequency of pain and the intensity of pain, measured in across different parameters. So again, very, very exciting for us and everyone who's been working with the program. Again, the patient community and the physician community as well. So partial lipodystrophy, this is a second potential indication that's being evaluated, and that phase 3 study is ongoing. A very different disease, but hypertriglyceridemia is certainly part, but one of very many complex metabolic complications that are involved in partial lipodystrophy, all of which result in a variety of significant medical challenges earlier in life. For example, premature heart disease, premature liver disease, et cetera. And at the core is both a fat storage disorder, but also a disorder of extreme insulin resistance that wreaks havoc across virtually every organ system in the body. Here's Andra, who is here this year as well, and is a tremendous leader of this patient community, and someone, again, we've been very privileged to learn from over the last 2 years. So this study is broadened, it's ongoing. We're continuing to enroll. It's been very exciting to work with. It's another case, again, how it wasn't part of the original development plan, but after the original results, it was the physicians we're working closely with who understood the patients who said, this is an opportunity that should be looked at. And over the course of this program, too, we've continued to learn and made adjustments actually to the clinical protocol, actually based on that patient input and the patient partnerships that we have at Exia. So very exciting, looking forward to following this as it moves along and we continue to work on enrolling it. So based on this, and this is really a setup and a tee up for the next panel, you might be thinking, if you're working on a disease that is not yet being studied with Antisense, what would be a good fit? Here's a couple of the lists, but I'll really turn it over to the next panel, the next session, where they'll dive in deeper, and Sarah will as well. So first, severe unmet need, of course. Sarah described how we really feel a responsibility to prioritize those areas where there's clearly unsatisfactory or unavailable treatment options, just as I described with FCS and FPL. Second, a patient population that is ideally identified and well characterized. That provides a path for the regulators. Disease animal models that either exist or can be developed, because that provides also a path forward for the studies that Sarah outlined. Available biomarkers, so a way of measuring those disease or other surrogates for disease progression. Natural history data, this is obviously very challenging in rare diseases, so even if it doesn't exist, perhaps there's an existing community that can be partnered with to obtain that natural history data that's very important to regulators, but also to the market access or the reimbursement authorities who want to understand what truly is the difference that this therapy makes. And lastly, a cause that is well understood. So we started out this presentation outlining there's the genetically validated targets, and that certainly makes it, that provides the key, if you will, to unlock how to proceed with antisense therapy. So to learn more about next steps, and obviously we're going to be taking questions here we want to hear, but the next session led by Sarah will get into what you need to know before approaching industry and really how to engage about the opportunity for a technology like Antisense to support and see if it can possibly help a different disease as well. So with that, Tammy, I'll turn it over to you. Okay, thanks.
I think I'll have you guys keep the mic to answer the questions.
Sure.
So if anyone has any questions,
sit on the chairs?
I can bring the mic around. I think I'll start with our first live stream question, which is for Sarah. There's a question about, can you please go over beta thalassemia and Ionis TMPR SS6IRX, and how will it help patients with beta thalassemia?
Yeah, so the program's just gone into the clinic. So we've just started the phase 1/2. The idea is actually both to help from an aspect of transfusion dependence, but also because of a mechanism of action. One of the things is that a lot of patients develop a buildup of iron and an iron toxicity, and to also be able to benefit that at the same time. So it's looking at a dual kind of mechanism approach. Of course, it's early days, as we're just putting the drug into phase 1. And in the phase 1, what we'll do is actually look to identify what would be an appropriate dose to be able to move forward.
Great, thank you. Does anyone in the audience have questions? Here, let me grab. Actually, I'll let you keep this.
Okay.
We get another mic, if we can. Thank you.
Moving mic.
Hi, I apologize if this is a silly question, but I was just wondering, how is Antisense, this technology, different from AAV RNA gene therapy? Or is it the same?
Yeah, so it's different from an aspect. When I talked where you had the DNA, you have the RNA and the protein, we interact at the RNA level. The AV9 vector is a vector that's often used for gene therapy, and gene therapy is typically acting at the DNA level. So it's a different approach and a different type of mechanism. I think from a gene therapy aspect, as human beings, and I know for me, I'm cheering on gene therapy. It would be tremendous if we could actually develop the ability to change it at a genetic level, but that's proven to be very difficult. Science is hard, and that's certainly very challenging. So we've got quite a long way to go from that perspective. What Antisense allows us to do is to be able to alter protein production, but to do it in a way where we're not actually changing the actual DNA. So it's in many ways, I hate to say it, but it's probably a simpler approach and a much more straightforward approach. Christina, would you add anything to that?
Okay, yeah.
First of all, congratulations. I've been in drug discovery for a long time. This is not easy, and it's amazing. I have a question for you. Could you share a little bit whether you have any experiments? Let's say that there is a foundation that fulfills this, that ticks the box of all the things that you say. Do you have any experience already working with foundations, and how would such a collaboration model work in practice? Or are you envisaging having collaborations with foundations who can tick your boxes? What is kind of your philosophy here?
Sure. Why don't I start with that? And so actually a lot through Christina is we get approached all the time. And our scientists, we have incredible scientists who are very active in the patient community, and we are always looking for ideas from a new target's perspective. In many ways, that's one of our limitations. We need targets to come in, and we have our program Inotersen as an example, which is for TTR amyloidosis. How that program originated was by someone external from the company basically coming to the company and saying, you should really look at this, and championing it and driving it through, and it then ultimately became a program. We're actually doing, I'm working on something at the moment with, actually almost at the other end of the spectrum, where a set of parents have, that their children have the disease, they've identified a target, they've actually funded a lot of the early research work, and are now actually at the stage of forming a company. And we've helped them from an aspect of helping with some guidance around VCs. And then, ultimately we're looking to see if there's a way whereby we can collaborate and partner with them. So it's kind of different ends of the spectrum.
Yeah.
So you're not really collaborating, there is no kind of let's call it now, business interaction between certain medical foundations in your company?
Not yet, no.
Is that something that you would be open to?
Potentially, yeah, absolutely. Absolutely. We were sort of from a philosophy perspective, were grounded on two things. One, that sick patients depend on us, and we're grounded on our science. And if there's something which looks like it would be very applicable to our technology and it makes sense with where we have expertise, then yes, absolutely.
I would just add to that 2 of the 3 targets that Exia is focused on both came to originally Ionis through outside partners who were lipid expert physicians who said, this is a really important target, you should look at this.
Yes.
I think the patient foundations, and I now can specifically speak for our own foundation, but this I think is much bigger, because I think we have that value of being that connector to both the research community.
Absolutely.
Oh, yeah.
Become extremely complementary.
Absolutely.
We kind of focused on the patients, you kind of focused on the drugs, and I think if we can have some, and I'm thinking now also we have to maintain these platforms, right? So we can start thinking a little broader, like almost like business interactions between.
Yeah.
You focus on that part, we'll focus on the rest.
Yeah.
I think we could some amazing strategic partnerships here.
Yeah, absolutely. And for us as well, when we start with a program, the better the understanding that we have about the disease that we're trying to target, the better job that we can do. And we have made mistakes, a lot of them. We've been around for 26 years, so we've made a lot of mistakes along the way. But one of the things that we're really looking to do more and more is make sure that we really understand a disease from a patient level. Because we have, and I will put my hand up, we've designed studies and used certain endpoints that, asked patients to do a lot. We had one program where we asked patients, almost it was an entire day. Now, one of the core aspects of the disease was fatigue. And we were asking these poor people to do these tests over the course of an entire day. That was a really bad idea. And we adapted as we went along, but had we known more of that up front, we could have done a much, much better job.
Yeah.
So if you could expand a little more on what fits well with Antisense in terms of specifics to certain diseases and kind of checking all those boxes, but would it work with structural proteins, gain of function, loss of function, deletion, substitution, put a little more parameters around whether it's an approach that you think might work in specific situations such as our disorder, KCNQ2, which is a potassium channel disorder, is structural protein. Some of the patients have gain of function, some have loss of function, deletions or substitutions, of whether this is something that should be considered.
Yeah. I think in terms of where our technology works at its very best is for a monogenic disease. So where there is a single mutation that we're targeting. SOD1 is a gain of function program. So we've done gain of function. Have we done loss of function? I know we've done gain. I don't know if we've done loss. Yeah. We've done in terms of there's like the traditional taking out a protein or upregulating a protein. That's kind of one of the things that we do a lot. We've also done alternate splicing. So it's not necessarily that limited. The limiting factor is more on understanding the disease at a very precise genetic level. Because if we have the wrong genetic target or where there's more than one, then we can go after one, but if there's something else going on as well, then obviously we're not going to be as effective. We tend to, when we look at programs, it would be very unusual for us to do a program where there's a small molecule already available or a monoclonal antibody already available, and where it's more applicable to that technology. For that, we'd probably say, you know what, we'll let others do it, and they'll do a tremendous job, and that's not something that we would do. Christina, would you like to add at all?
Really important to know what the target is and to know the pathway involved with the targets and the other targets that are involved in that pathway. It's more of a knowing what the cause of the disease is, and then the different paths that lead to the disease, because there are multiple areas along that path that we can actually target. Sometimes you may think it's a certain specific target, but it's actually a different downstream target that you actually want to knock out to help the disease. So there's a lot of opportunities.
That's a great point. Just building on what Christina said, that was the experience with Falada Source Sin, right? Because we're actually not targeting the underlying genetic mutation of either of these diseases, but a parameter which is an important driver of the pathology itself.
I think we've got another question over here.
Well, you kind of answered my question. So I'm Les Rogers from the Cardiofaciocutaneous Syndrome International, where restopathy combined, it's about 1 in 1,000 births, NFs over there. So we have a pathway with multiple genes, and I guess it kind of gets to what she was getting at, which is, the question is, do we target an upstream gene or do we target a downstream gene? And I don't know, you kind of answered it, so she walked over afterwards, so I'm going to ask a different one, which is, how much does the process cost, and what's your model for the financial side of this?
So, it really depends on a program. From a cost perspective, we are efficient because we're using the same technology, that there's some things that we don't have to do. From a philosophy around partnership, so Ionis is different from a lot of companies in that we're not a fully integrated pharmaceutical company. So Ionis as itself we don't commercialize our drugs. We commercialize our drugs through examples like where we set up a company, like Exia, or where we partner with companies who have direct commercialization and late-stage development expertise in that space. And a lot of those, how the economics of those agreements work, depends on where a drug is in development. As to sort of the types of royalties, the types of upfronts, it really varies as to how far along you are in the development process. But we have a whole range of different types of collaborations. We've done sometimes, we don't do it very often, where we take stock in a company. So it's really varied. I think every type of possible business agreement and type of transaction structure we've probably done.
Are there any other questions in the audience? Okay. Thank you both very much. Let me check our live stream. I don't think there's any more on the live stream, but I believe you can continue to submit questions that I'll monitor.
Actually, can I just before we close out, because I realize I didn't address one aspect of your question when you said how much does it actually cost? It's hard for me to give a specific, but one thing that probably I could say is how much money we've spent since the company was formed, and I think it's about $3 to $4 billion so far. But that's on a whole range of different programs and some challenges along the way.
Thank you. So thank you both, Sarah and Molly. At this time, we would like to ask you to jump onto the mobile app and take our brief survey on this session. And then we have a few minutes, about 10, 15 minutes to transition to your next breakout session. But first, let's give one last round of applause for our two wonderful speakers. Thank you.